So those changes you're talking about, they're going to have to be obviously significant enough to create true novelties and innovations. So obviously just shuffling of this existing information, it's not going to produce a new genome or a new code from that English book. I mean, it's for example, E. coli, Lenski's E. coli experiment. Bacteria have very short generation times. They've been doing this experiment for years. How come they haven't gotten anything other than a bacteria? If we're going to see large-scale evolution, new information, an English code to a German code, why don't we see that? It's not.
That's not an expectation for things to be their domains.
And English code to a German code.
May I?
Yeah, sure. I'll work off it's fine.
Sam, I don't want to step on your toes, bro.
Well, no. I mean, like, listen. If we want it to just be a group discussion, he wanted a one-on-one. Like, I'm just getting kind of confused by it. I'm not frustrated.
Yeah.
No, I'm frustrated.
Yeah, I'm just going to have to take you.
If I see populations of dogs producing other dogs, and then you say a population of dogs today came from a population of non-dogs long ago and far away, but yet you can't show me or provide me a mechanism of how that's possible, I'd be frustrated too, guys. Ponds come to people evolution. You can believe it all you want, but it's just not science. It's not observational, operational science.
Even read Nif, hold on a second.
The article.
Hold on. Question, please. Can I ask a question?
I'm not saying it does.
Please, can I ask a question?
Why did you cite it? Then why did you cite it if it doesn't?
Because this is the very mechanism that supposedly created the 80% difference.
Can I ask a question?
According to who? According to who? Not this paper.
Wait, you're saying it's not?
Question.
Oh.
Not according to this paper. That's not. This paper is not.
It doesn't matter what that paper says.
Well, why did you cite it?
What matters is okay. So let me ask you a couple of questions to get.
Oh
to
my
this.
gosh.
Okay. Are you telling me? Is it not true? Is it not true that 80% of the proteins in the human?
Can I ask a goddamn question? I do not normally get angry, but I am frustrated.
James, hold on. The difference. The problem is
not a conversation. This is a monologue.
James, the problem is this: the evolutionists, when you point out this kind of evidence, they go ballistic and atomic on you because they can't answer for it. And so that's what you're doing, and moving the goalpost endlessly. So I'm going to ask a couple of questions to get to the meat of it. And the first question is this: is it not true that 80% of the proteins in the human genome are different by one amino acid, right?
I cannot deal with. I am very angry. Let me tell you. Let me talk for 1 minute. Will you please let me talk for 1 minute?
Is that not true,
guys?
Will you please let me talk for 1 minute? You keep on saying you're going to let me talk, and that you never do.
And
the follow-up question. Please just let Arjun ask his question, please.
Is it not the same mechanism that created the difference in our proteins?
Oh my God! Please, shush. The other people who have their microphones muted, will you please tell us, do you think that Nephilim keeps asking the same question over and over again and does not allow us to talk about it?
The problem is this, all right, everybody, look, when you ask these questions, atheists don't want to answer them, so they talk over you constantly.
Just want to make it clear to everyone listening, like if we look at our model of pre-existing heterozygosity, would you say that science, you need to have testable and falsifiable predictions, right?
Sure. I mean, that could be done fairly easily, right? I mean,
I just want to let you, because obviously, we've talked in detail about, the non-protein coding regions, pre-existing heterozygosity, but we're saying that obviously this most of the non-coding DNA will be functional, okay? And obviously we would probably agree to disagree that what we're seeing is functional. I would say that it's clearly not idle, but here's the big thing, right? Differences in, say, mitochondrial and nuclear DNA sequences between families are expected to reflect functional differences, but not just due to neutral mutation. Okay, because if we believe in original created kinds, we're going to agree in function, okay, genome-wide function, but different functions across families. Because we would say that the created kind is, you could probably take it up to to the family level, but measured mitochondrial DNA mutation rates and known mitochondrial DNA differences, say within families, they would be expected to fit the creation timescale and what we would assume is a constant mutation rate, because the created kinds, and obviously if you understand the model, they're obviously going to start with no mitochondrial DNA mutations, right? Because we inherit mitochondrial DNA through what? Our mother, right?
Okay, I mean, I think even if you're going by, creationist standard, you would still assume that I wouldn't grant you that there's a constant mutation rate, because, and again, we seem to both agree to the fact that the environment can cause or can dictate the rate at which mutation can be affected.
But would you say it's fair that if evolutionists are allowed to inherit mutations, say as the source of all variety, why can't we as creationists inherit pre-existing differences? Like, would you say that that's fair if we're comparing models?
Well, I think you can, but I think that generates more harm than good for you. Because if you do genomic studies across species, you'll find that a lot of species, I mean, humans and apes, and there's quite a few, like mammals, for instance. We share quite a few different, we share quite a lot of genes, and some of them we don't even use. Some of them we really don't have a function for, but they're there because we call it evolution. You guys would call it, I don't know.
Okay,
but the point being that we share genes that aren't even within our own family sometimes.
So are you saying that the environment determining factor is natural selection? Would you be able to make a prediction based on natural selection? Because if we look at these constant mutation rates in regards to mitochondrial DNA, the differences would actually point more towards a 6,000 year time scale versus a 200,000 year time scale, say back to the origin of humans. Go ahead.
Right. So I mean, like, if I'm going to argue that, like, temperature, for instance, or the environment can play a crucial role within the mutation rates, I can subject mice to all sorts of different temperatures. I can have, some colony of mice that starts off, give them a very happy life, food and water and all the things that the mice needs. And, after a certain amount of time, I segregate them and I say, okay, so one of these mice are going to live in normal conditions that mice normally would operate under, but some of these mice are going to operate under conditions that would resemble more of a drought situation. So they get less water at certain increments. The heat, there's more heat that they're exposed to. They have significantly less rest time. I can create parameters to test, what are factors that could increase mutations within these mice.
Okay, so can you fill in the blanks, say, of how much natural selection then has to eliminate in order to, say, fit within the evolutionary theory? Like, would you be able to sufficiently fill in those blanks and then make that successful prediction?
I'm not sure if eliminate by meaning like silencing of certain genes, or if you mean just out and out eliminate, the gene itself.
Well, I'm just saying that it seems like there's a lot of guesses, which means no testable prediction, which would indicate that it's obviously not scientific. Because from my understanding, most nuclear DNA differences, if we're looking at, say, equids, between them, they're homozygous within those species. So if we look at the 26 million nuclear DNA differences, let's say between the imperial zebra and the domestic horse, just 2 million are heterozygous within the domestic horse. And we actually see that other species display this same pattern. So if we actually look at all the nuclear DNA differences, they're significantly more than obviously the mitochondrial DNA differences, and we would say that's because of pre-existing nuclear DNA differences, and we would say that the mitochondrial DNA differences are based on a constant rate since Eve, right? Because we pass it down from Eve. Go ahead.
Well, I guess to some extent that would hold. I mean, the first part of what you said I don't agree with, because the fact that, sure, it's hard, but That's also why people are invested in the field of science. I mean,
well, how much? Okay, well then I guess it's simple. Then how much mutations does natural selection have to eliminate then? Like, obviously, for it to be testable and falsifiable, and you need to fill in the blanks, and you need to make a prediction on natural selection. How much mutations does natural selection ultimately have to eliminate? Go ahead.
I mean, you're asking. Okay, so. In a controlled experiment, we may be able to test that.
Are you currently testing it, or are you saying that this is the prediction?
So what I'm saying is that, like, if I were to set up an experiment with mice under conditions that I knew, I could probably make reasonable predictions about how many mutations occur per generation or something to that effect. But for me to say, here's how many mutations will need to occur for, this mice to go to something that's, a different species or something like that.
Well,
I need sufficiently more data about the environment, about, how these mice were living, what kind of pressures are they under, things like that. I wouldn't have enough information to judge that.
Okay, so what you just said is you can't, and it's only a guess. Now, let's look at the family of obviously cattle, sheep, antelope, because this is one that we can actually look at, okay? Because they have been analyzed. So there are hundreds of species in this family for which there is ample nuclear DNA and mitochondrial DNA information for analysis, okay? Now, if we look at it, and I urge you to do so, there are only a few thousand mitochondrial differences separating any two species within this family, which is amazing, because what we do then is we draw a branching tree from these data, okay? Now, what the tree shows, it shows splitting, okay? So the distances between the splitting events, let's just say the branch points to new species, are reflective, obviously, of the number of mitochondrial DNA differences. Now, what I said earlier, we're assuming a constant mitochondrial DNA mutation rate, okay? Now, the plot shows the number of species as a function of time. So what the plot suggests, if you're following me, is that there is a constant mitochondrial DNA mutation rate within families, and the mitochondrial DNA mutation rate per species decreases as homozygosity increases. But for nuclear DNA, and I'm almost done, speciation decreases heterozygosity and hence the potential for speciation. So if we actually look at the species that have been analyzed, it's more consistent with our model of pre-existing DNA differences and constant mitochondrial DNA rates. Go ahead.
Well, sure. I mean, that's all well and good. The first question, of course, is, you're dealing with you're dealing with some amount of species. That's fine. Certain species of animals have different means of survival mechanisms. Some are engaged by sex arrangement. Some are engaged by hyperactivation of certain chromosomes. Of course, the thing that has to be asked is this constant just throughout this particular selection of species that were chosen, or is this constant throughout all species?
Well, obviously, I mean, we've only sequenced certain species. I'm looking at species.
Well, sure. I mean, I understand that. But of course, though, that's fine for the sample size that they have, but it's still speculative.
And that's our prediction, right? Because my whole point is mutation rates of a handful of species. The whole point of everything I just said is they are consistent. That's how creationists, but not evolutionists, can make predictions on all animals' mutation rates. So whether it's the pre-existing nuclear DNA differences that we say are based on pre-existing heterozygosity or constant mitochondrial mutation rates, we're making the predictions. And so far, the trajectory is pointing towards a stronger model based on a biblical-based model. And I understand that we'll see what the future has in store, but some of these testable predictions are coming true, which I'm actually posting in the description box. But it seems like the trajectory is stronger for us than it is for evolution itself. Go ahead.
And I think a few evolutionists have suggested the same thing, that mitochondrial DNA differences may be the contributing factor towards speciation. The question, of course, is because it's young, how much of a bearing does it have on in the rate of speciation? I mean, it could be significant, but it could also contribute 1%, which in that case, that's fine, but that means there's something still larger at play.
But if the DNA differences, say, of all species so far, obviously we haven't sequenced them all, if they're too few based on everything I'm saying, evolutionists have always predicted far too many differences. So why is that the case? And after you explain to me why that's the case, would the rescue device be natural selection? And if it is natural selection, I want a testable, falsifiable prediction as to how many mutations natural selection has to eliminate. And if you can't give me a testable, falsifiable prediction, then it's not scientific. Go ahead. Because it sounds like we're agreeing that based on these mitochondrial DNA differences and nuclear DNA differences, the creation model is the one making the testable, falsifiable predictions, and the trajectory of the non-coding protein regions is pointing towards genome-wide functionality. Now, obviously, we'll see what the future has in store, but what I'm saying is, based on all this current evidence, the creation model appears to be more scientific. I just want to know where this new information is coming from, not a shuffling of existing information, because you could have a dog population producing more dogs, but you're always going to get a dog.
It comes from environmental pressures. You know what I mean? The environmental pressures is what is what cause the new information, quote unquote. That's another word that's loaded. To appear.
Well, I mean, it's just a shuffling of pre-existing information. So obviously, if it's like, for example, the wingless beetles, on an island, they lost their wings, and now it's beneficial because in their environment, they can survive because the wind's not going to blow them away. But at the end of the day, it's still a loss of information. They lost the information for their wings, so that's still not going to take your fish from to fishermen. Go ahead. Well, that's the discussion, right? The biblical model, we would say that God created, pre-existing differences within all the created kinds. He created significant heterozygosity, right? So with all that allelic variety, we could see a ton of variation based on mutations and natural selection. So we don't need to see new novel information, but evolution requires new novel information. So,
okay, have you ever heard? Hang on. Have you ever heard of the game of life?
Yes, I have.
Okay, now, based on what I remember, is that complexity arose just from a few simple rules that the guy made. So why couldn't complexity arise from simple simple atoms to hold on. Being affected by the four fundamental forces of physics?
Well, I mean, yeah, you can believe that, this evolutionary
hold on. We have, I would say the game of life is kind of a good evidence for that. Like, there were simple rules, that the programmer made that govern the game or the interactions or whatever, that gave rise to complexity.
I'm sure it was based on very specific programming and I'm sure there was an information system that this game of life, this program, was following. Obviously a single-celled organ, because technically for evolutionists, like you're saying, which is good, the ultimate cause of genetic change is mutation. But what I'm saying is the mutation rates, they're going to be too slow, which means the number of differences, say, in the organisms, they're large. Okay, so that's it's too, it won't explain the current diversity. But my point is, with us creationists, like I said, heterozygosity, meaning millions of differences in DNA sequences of chromosome pairs, that was built into organisms from the beginning. So long periods really aren't required for these new species, and that explains all the life we see, this pre-existing heterozygosity. But how do you explain all the life we see? Because I get your analogy, but if it's just acting on pre-existing information, I don't understand how you're going to get your fish to fishermen. Go ahead. Because if you think about it, starting from heterozygous organisms, you know, visible distinctiveness in offspring, it's going to be apparent. So if you look at recombination, gene conversion, that's going to provide many new varieties of combinations of traits. Are you familiar with Mendel's P experiment?
Well, what? No, explain that. What is that?
Well, because this was around the time of Darwin. So Darwin wasn't familiar with genetics, obviously, right? So Mendel, if you look it up, he and some of the people.
Wait, hold on. I remember now. Maybe you pronounce it was the sorry, give me a sec. Yes, Mendel is the one.
He was looking at traits. Yeah,
plants.
Yeah. So he noticed that these traits, they remained distinct, right? So they didn't blend. But the thing is, what he was looking at and what he's seen was, you know, recessive versus dominant. So they appeared and disappeared in family trees. You know, they came in these dominant and recessive types. So at the end of the day, Darwin, he didn't understand genetics, but now we see, say with heterozygotic organisms, you know, you can see a variety of traits in a short amount of time, and he's seen that with these peas, these plants. And I don't even think Darwin was aware of Mendel's work. And the whole point is it shows that, you know, new varieties can come about from pre-existing heterozygotic organisms and change. So that's consistent with creation. I mean, do you believe this type of change is not consistent with biblical creation?
At the end of the day, dude, I don't know. That is the honest truth.
Well,
if you don't, you know, if you can't respect that, then I don't know.
No. I completely respect. I think you're very open-minded, but a lot of evolutionary apologists and defenders that I talk to, you know, they're not exactly open to, I guess, another, say, model, because we're trying to say that this change is due to a sorting out of information, not a gain of information. But evolution would need a gain, or, you know, the addition of new genes, say, to encode for heart, lungs, brains. Like, I just don't understand how that's possible. Can you explain it to me?
Nope. At least not currently.
So technically, you know, if dogs produce dogs and elephants produce elephants, that's what we know to be true. To say that they share a common ancestor, that's not exactly science then, given what we know about empirical science, right?
If you say so. Like I said, I don't know.
Yeah, so I'm just saying that even when I talked to Drag, and he left the conversation very shortly, if we look at genetic entropy, which is that accumulation of the near neutral mutations over time, you know, that's information degradation. So I just don't understand how you're going to get rich by losing money. And evolution would, you know, predict these, you know, from simplicity, like you said, with the game of life, to more complex organisms. Is that what you would assert happened in the history of life?
Well, I don't assert it. If there's evidence for it. I mean, you know, the four fundamental forces exist. I mean, we have gravity, the weak and strong force, and electromagnetism. I mean, shit.
You're gonna get some type of function based, because remember we were talking about spurious transcription.
Yeah. Well, but what I'm
the ERV.
So what I'm talking about is that the ERV, in order to produce the viral proteins, it's got to transcribe them, right? First, it uses reverse transcriptase to get its genetic material into the genome, right? And then it has to transcribe its genome back out so that it can be translated. So that means it has to have a bit in there that will induce transcription. Now, if you lose the viral genes, but you keep the promoter region, you just get transcription of whatever is left.
Okay, so where I'm coming from is I'm saying, and obviously I'll allow you to respond to this, but I'm just saying viruses, at least it's not observed, they do not randomly insert tens of thousands of, say, these endogenous retroviruses, which just happen to be in exactly the right location and having exactly the right sequence to perform some valuable functions. So I'm saying no ERV should have function, because the fact that there's increasing evidence that they have important functions in the placenta and in the embryo, and the fact that they aid transcription in a fifth of the genome, I would say this is strong evidence that ERVs were possibly not the product of retroviruses, or it could be a weakness in the system. But most importantly, and you're going to know about this because you're a scientist, there are cell processes designed to weed out harmful or viral infected genes known as apoptosis, right? So why, after millions of years, do we still have so many viral infected ERVs, ERV cells? Because shouldn't apoptosis have ridden them long ago? But apparently it hasn't done anything at all. Go ahead.
Well, that's not how apoptosis works. But anyway, so you previously defined functional as basically transcribed, right? But any ERV must cause transcription.
Transcription, but also very important placental roles. And
well, no, but you didn't define functional as being transcription plus do something. You defined it as just transcription.
Do these ERVs do something?
Some of them do, but that's not how you define functional. You define functional as just transcribed. If it's transcribed at all, according to you, according to what you've said multiple times, including more than once in this hangout, if it's transcribed at all, it's functional, according to you.
Transcribed suggests function. We haven't done these gene knockout tests.
Well, actually, it doesn't suggest function.
Well, you just said that these ERVs have placental functions, embryological functions. That's consistent with my model. You can have any type of interpretation of this all you want.
It's not consistent with your model?
It's consistent with design.
But, so firstly, the fact that some ERVs have some functions which may or may not be necessary, depending on context, does not mean that all ERVs are functional. Obviously. Additionally,
I think it's important for the audience first, because I believe we speak, and by speak I mean understand, maybe 1 to 5% of the DNA language. How much do you believe we understand or speak of the DNA language? Go ahead.
I still honestly don't know what you mean by the DNA language.
Okay, so for one, we've already overturned ERV from junk to functional. Okay, so I think that's consistent with us. Secondly, my question is, once again, I'm going to state, how much of our DNA, deoxyribonucleic acid, how much of our information systems do we truly understand? Like, another way to ask it.
That was a remarkable restatement of the thing I asked you. You just re-said the thing I asked you.
You don't understand such a simple question. If we perform these gene knockout tests on humans and other mammals,
because it's like
what type of functions they have, why are you so?
How much
we understand it all?
How much of the squanch language do you understand? What the hell are you talking about?
Exactly. If I look at a Russian,
I know what DNA is.
I'm not gonna understand any of it. But it doesn't mean that if I look at something that I think is junk or non-functional, I'm not gonna conclude, oh wow, that really is junk, or an evolutionary leftover, or an ancient repetitive element. I'm ignorant of the language, and I'm saying you're ignorant of the DNA language. That's all I'm saying.
Oh, so yeah, we can tell whether or not things are functional.
Oh yeah, have you done these gene knockout tests on humans and on other mammal species? And how much testing have you done?
Funnily enough, yes, I have. But there are other ways beyond knockout experiments to test for functionality. And knockout experiments aren't even really the best way to do it necessarily. So
I would disagree with that, but go ahead.
Well, I'm really impressed that you would disagree with it, but I don't know what your point is, because, like...
Okay, about the, say, embryonic or placental development, okay? I would say it's a mystery at this point. The fact we don't know the manufacturing tools of, say, embryonic development does strongly hint that we don't understand most of the DNA language. And the fact that we only speak 1 to 5% or understand 1 to 5% of the DNA language, and ENCODE has revealed that 80% is transcribed. And all I'm saying is that suggests functional. Okay, so that's our prediction.
So when you say that it's found that 80% is transcribed, you understand that all that means is that they got at least one read in one sample, right?
One read in one sample. Yeah, obviously I understand that, you know, that this isn't. How much did ENCODE actually test?
Well, they tested a lot, but the question is, if you only find one read in one sample, when you would expect for something that's actually doing something that's being transcribed beyond a spurious level to find millions of reads across the samples. Do you think that one read in one sample is impressive?
I would say that doesn't definitively give us a function, but it does suggest possible functions. So I want to ask you, pseudogenes, are they part of this 80% of that section that ENCODE tested? Would pseudogenes be part of that? That was shown to have this spurious transcription or transcription?
I imagine they tested a lot of different things.
Okay.
It's been about 3 years since I read through their work in detail, so
okay, well, I'll give you the answer. Pseudogenes were definitely part of the 80%, and we know that some pseudogenes are actually no longer pseudo. That means that 80% function is beginning to look more and more reliable as we are predicting. And the fact that we know so little about the embryological developmental phase is strong evidence.
The real problem is that we know that the absolute maximum amount of the genome that could be functional is 25%. At
you know that for a fact?
Most, yes.
So you speak 1 to 5% of the DNA language. You know nothing about the construction tools that are taking part in that developmental phase.
We actually do know 25% a lot about development. I don't know where you're getting that we don't know anything about development. We know a lot about development.
But the thing is, a lot of this development, a lot of these construction tools being utilized during that phase are in that non-protein coding region that we, or evolutionists, might say are junk.
I can't imagine what your point could possibly be with that.
The whole point is the trajectory is pointing towards, I understand you disagree, it's pointing towards genome-wide functionality, but based on the fall, based on our model of degradation and genetic entropy, which I understand you don't agree with, that's fine, we will expect some junk, some broken genes, some pseudogenes, but not, say, as many as evolutionists would predict. So I feel the empirical evidence is more consistent with biblical creation. Go ahead.
Yeah, well, but the problem is that, again, there have been studies done, fairly substantial ones, that have demonstrated the amount of the genome that could possibly be functional, and this is on a very broad, both theoretical and experimental basis. So, if you're wanting to say that it's, you know, the majority of the genome is functional, you're already wrong. You just cannot be right about that because it's already been demonstrated to be false. Now, I know you're not up on the literature.
Oh
but
no!
Actually,
I'm up on
that was demonstrated like 40 years ago.
Evolution predicted junk since the 70s. Would you agree with that? Since the 70s, they have predicted junk. They didn't predict.
Yes, 1972.
They didn't predict that ENCODE would sequence and look at that analysis there and conclude that 80% is transcripted. So now you have to use your rescue device based on that evolutionary bias and assumption and say, oh, well, it's spurious transcription. Oh, well, it's this, it's that. Common ancestry markers in DNA has a junk DNA assumption to work. That's a fact. Without junk, you're genetically dead. And
are you under the impression? Under the impression that spurious transcription is a concept that came about after ENCODE?
No, I'm saying that they didn't predict that 80% of that would be transcripted. There's
actually the point of spurious transcription is that it predicts that the majority of the genome will be transcribed at a spurious level.
Are you familiar with Francis Collins, who's I think he headed either the Human Genome Project or, I mean, he's pretty
yes, I know who he is.
Okay, and this definitely isn't a quote mine, but even he said, and I think this was after his prominent work with Language of God, and a lot of evolutionists look to him actually for evidence and information, but he said in terms of junk DNA, we don't use that term anymore because I think it was pretty much a case of hubris to imagine that we could dispense with any part of the genome as if we knew enough to say it wasn't functional. Most of the genome that we used to think was there for spacer turns out to be doing stuff. Now, wouldn't you say phylogenetic systematics relies heavily upon non-functional nuclear and mitochondrial DNA differences? Like, isn't that a primary assumption? Because that all goes back to now, the junk DNA argument. That also goes back to these differences we're talking about.
We can include that if you want to, but that doesn't change the result.
Yeah, but I'm trying to say, if you're looking at these genetic markers and assuming that they're evolutionary leftovers based on past common ancestors, and we're showing that these are actually functional differences based on pre-existing heterozygosity, that means these phylogenetic charts are based on a primary assumption that molecular semantic evolution is true, but now we know it's not true, so now these phylogenetic charts are just lines on paper. Go ahead.
No, so none of that needs to be assumed. What needs to be assumed is that certain genes that exist in the past have some distinct function that arose from previous genes that we can find within organisms, and that we can map and somehow determine a distance and somehow an age for these organisms and for these genes that we're observing.
As he said, those are just lines on paper. That's what it comes down to.
Because the whole point is, if they're functional, they now become a common design argument instead of a common ancestry argument. So just like Warning said, now they're just lines on paper, and lines on paper aren't evidence for anything, really. And I understand that if they are just plagiarized mistakes, because I understand the evidence for evolution very well. And yes, plagiarized mistakes would point to universal common ancestry, but if they're functional DNA differences encoded from the start, then no, that's evidence for design. And that's a prediction made, by our model. Go ahead.
Well, I mean, it depends on what. So again, for something to be functional, it has to have something that can cause it to function.
Well, do you want me to
go in the ground? That's not going to work.
I've already told you that, expressive, regulatory, embryological, placental roles, these are functional roles. And clearly ENCODE has revealed that 80% is transcription. I understand that you say that that's spurious transcription, but it proves that they're not idle. And it proves that we know very little about the genome. For example, protein moonlighting is something that was newly discovered, not too long ago. And that reveals that proteins have multiple functions. And the more we learn and study the genome, the more we realize just how complex and functional it really is.
Of course. I mean, that's not surprising.
Well, that's evidence for
multi-dimensionality.
No nucleotide in the genome is idle anymore. That's a fact. So spurious transcription and just labeling all these important functions that I've stated as not really functional at all. I mean, I see that personally as a rescue device because idle was the basis for junk DNA in 2001, but now we know they're not idle. So now the theory has to be adapted to fit the data. There's always a lot of adapt. Let me ask you, how many vestigial organs were listed 100 years ago? Wasn't it like 140 or something?
I don't know, probably.
It was like 142. And now there's about 5 or 6 arguable ones that I would say aren't vestigial, but that's a huge difference. So that's just based on ignorance, because obviously they didn't realize what things like our tonsils and our thyroid and obviously the appendix and coccyx and all these different things were for. But now they realize that these things actually have important functional roles, and now they're not listed as vestigial anymore. So how do you feel about that?
Well, it's based on existing data, just the way Hes Mendel tried to prove with his own experiment.
Exactly. So
people make wrong conclusions. That's perfectly fine. That's how scientists learn.
So what makes you think that these vestigial genes or pseudogenes or junk DNA is not exactly the same thing as the ignorance of the vestigial organ era? It's the same thing. I mean, I don't see why you don't see that.
No, because I would see that, if a gene is indeed functional, if it's non-vestigial, we should see it being transcribed in an organism at a relatively at a regular
80% transcribed.
Sure. I mean, how much of that is spurious transcription now? Once again,
I would say very limited of it, based on all the important functions that I've already listed, and I can certainly.
Would you consider one transcription event out of like 80,000 to be rare?
Well, like I said, no nucleotide in. But I don't believe that that's even the case. Because I'm saying no nucleotide in the genome is idle. I'm saying it's all functional minus a few pseudogenes based on genetic entropy. I mean, that's our prediction, and it seems like we're the ones that are making the predictions. It seems like you guys are just coming up with all the rescue devices based on all the current literature. If you could explain to us what about the phylogenetic systematics, what about genetics, genetic similarities demonstrates universal common ancestry. Go ahead.
Phylogenetics demonstrates that the bigger the difference between you and some other animal, the farther you will separate it. So the longer you will separate from the common ancestor. So compare a human genome to a mouse genome, they would have been the last mammal split off, because mammals were originally mouse-like, and then they split off and stuff. So the farther apart the genetics or genetic difference, then the farther your ancestors splitted.
Now, and that's and that's a good point. Now, are you familiar with the fact that phylogenetic systematics, as you're talking about here, it's mostly based on the concept of outdated junk DNA? So since functions are only beginning to be found, say during the last decade, meaning we actually understand less than 5% of the DNA language, the trajectory, and I will also say that a lot of the data is preliminary, but the trajectory is most important. It strongly suggests that the vast majority of DNA will be functional. Now, why that does not bode well for phylogenetic systematics is because this takes away the primary assumption of evolutionary phylogenetics, which is actually based on mutations for one, and pseudogenes, right? Genetic markers. But if the vast majority of DNA is functional, as we're seeing now, Godless Galaxy, it now becomes a common design argument. And what's nice and revealing is that creationists have always predicted that the vast majority of DNA is functional. So this functionality is certainly a problem and inconsistency for phylogenetic systematics. What do you think?
So you're saying that because we do not completely understand each function of DNA, that's like one of the reasons why we can't use phylogenetics?
Well, because a lot of these, phylogenetic trees, they're based on genetic markers that are assumed to be, mutation related, or let's use pseudogenes, for example, plagiarized mistakes or what they would call them, right? And if they really are plagiarized mistakes, that would certainly be strong evidence for universal common ancestry. But in regards to the ENCODE project, they found out that at least 80% of the non-coding DNA was actually transcribed into RNA, right? So that actually suggests function, but it's also been found that these non-coding regions, and most of our genome is actually non-protein coding, I'm sure you're aware of that, but it's now known that these regions of genomes correlate with biological complexity even better than the protein coding regions. So that actually suggests that these non-coding regions, Godless Galaxy, they carry significant information, say for regulatory activity. But a big thing as well is ENCODE. They didn't look at the portions of the genome involved primarily and exclusively in development. So that type of DNA would only have function then. So that means the trajectory of discovery favors genome-wide functionality very strong. Probably, there's certainly a lot more function in the genome than evolutionists formally assumed. But the point is if these genetic markers are actually functional DNA differences, they can't be used as plagiarized mistakes to build these phylogenetic trees. That's the point, you know what I mean? That's the point. So now these trees just remain in the realm of hypotheses. Right now, that's why the battle, rages on. Did ENCODE really detect function, or is it just spurious transcription? But if it really is function, once again, genetics points to common design. Like, do you understand what I just said there?
Sort of. So you're saying because these markers, they will claim to be mistakes or whatever, but that's a way to disprove evolution?
Well, because a prediction based on, pre-existing heterozygosity would be that the non-coding regions are functional, okay? But evolution says that there will be much non-functionality, so they use a lot of those, say, pseudo genes, which are actually in those areas, as genetic markers to build their trees. But like I said, in regards to ENCODE, the ENCODE project, these results have suggested that at least 80% of the human genome is functional. But as I said, is the results are preliminary, right? I've talked to scientists on this. I wouldn't want to straw man the, ENCODE and what some of the evolutionary scientists are saying. They'll say that a lot of it could possibly be spurious transcription. But one thing is clear, is that, most non-coding DNA is at least not idle. It's doing something. Now, why would the cell be wasting so much energy transcribing if it's not really doing any useful activity? So I think that's kind of a rescue device on behalf of the evolutionists. But I think the whole point is, if these pseudogenes, if these genetic markers that are being used to build the phylogenetic trees are functional, then that means these phylogenetic trees are just based on a false assumption that molecules to man evolution is true, but now we know it's not true. So they really are just lines on paper, and lines on paper don't prove a thing.
I'm going to explain what the Encyclopedia for DNA Elements, the ENCODE project, means for human genetics with the help of my trusty ping pong balls, some string, bit of tape, a few paper clips, and some cherry tomatoes. Right, here we have a section of human DNA, the human genome. The blacked out ping pong balls are bits of DNA that we previously didn't know what they did, and the red bits here by tomatoes are genes. Now, a decade ago, the Human Genome Project gave us a complete list of the 3 billion letters that make up the human genetic code. And people said at the time that we had the Book of Life. Now, it turned out it was harder to read than we expected. When we looked at all these genes and all the DNA, scientists found only 20,000 genes, and that makes up about 2 to 3% of all our DNA. The question became, what does the rest of this DNA do? In this model, the black balls. It was mysterious, people didn't know what it was. They actually called it the genomic dark matter, the dark matter of the genome, or worse still, junk DNA. For a while, scientists weren't sure if it did anything at all. Thanks to the ENCODE project, we now know what most of our DNA does. These newly revealed regions, the white ping pong balls, these are sections of DNA highlighted by the ENCODE project. Their role is to control what happens with our genes, and in this analogy I'm using the cherry tomatoes. So the majority of our DNA is not making proteins to build our cells and such like. What it's actually doing is controlling how these genes work. That might mean they're acting as switches to turn them off and turn them on, and where and when in the body that happens, or they may act like volume controls, turning a gene up, turning a gene down a little bit. So what use is all of this? Well, first off, it will help scientists understand how our genes operate in our bodies. These switches are incredibly crucial to what genes are doing in our bodies, and they will make the difference, for example, between how our body makes a liver cell as opposed to a kidney cell. Perhaps more importantly, the ENCODE project will help understand what goes wrong in scores of diseases where there's some genetic element. Now, scientists have already used the ENCODE data to identify 400 of these switches in the genome which feed through into diseases and other medical problems. And that is surely just the beginning of it.
I guess if you had to just throw out a a couple really solid examples of, say, new novel information, meaningful information being added to the genome that's not deleterious in any way, because obviously you're not going to get rich by losing money, what would you say that example would be? So I understand there are some good examples, but what example would you provide?
A beneficial mutation that is today?
Yeah, like a beneficial mutation that actually adds new information to the genome.
There's the ApoA mutation from the ApoAI gene. It's the gene that it happens in.
I just want to be on the same page. Is this the Milano protein, or is this the Milano mutation?
Yes.
Is that where like one amino acid has been replaced with a cysteine residue in a protein that normally assembles, say, high density lipoproteins? And obviously we know those are involved in what? Removing bad cholesterol from arteries. Is it that one? So it's kind of better protecting against atherosclerosis?
Yes.
So if we actually look though at the mutant form of the protein, it's actually now less effective at what it was supposed to do, because now it does, I agree,
but the thing is it's not necessarily, because this particular gene does that function, and it does the mutated version of it does lose effectiveness, but it doesn't completely get rid of it.
Okay, so would you say that it is, in a way, a loss of specificity? Like it's a loss of specificity that is now resulting in an amplification of a pre-existing gene or protein, I should say. Because, in other words, the specificity, as you were saying, of, say, the antioxidant activity for the lipids, it doesn't really lie with the mutation itself, but with the protein structure. But that already existed. So it's just an amplification on pre-existing information. But since we predict pre-existing heterozygosity, pre-existing DNA differences, it's just a trait being revealed or amplified based on the pre-existing information. So it's not really anything new, novel, and meaningful. Go ahead.
Okay, well, for starters, I'm going to need some kind of source for this claim here that you said that about it, because I would love to look it up myself.
Well, you just I just, correct me if I'm wrong, didn't you admit that the protein has lost a lot of activity for making HDL? So now the mutant protein has sacrificed.
I didn't say that it loses a lot. I said that it might. I mean, because I don't personally know much about that as far as it goes of that. I just know that it is indeed still a beneficial mutation because of the added function that it does.
No, and
any source that you have, I would love to look at. That's what I'm saying.
No, and I'll send you the sources and the articles that I've studied and looked into. But I'm just saying that the fact that, say, whoever these people are, I think it's a small population with this specific mutation, they're obviously unable to produce the normal levels of HDLs. Okay, so that is good, but we know that those are known to perform, and you might be aware of this, a valuable role in moving the bad cholesterol. So that suggests there could be, and you also said there could be, a health downside with this mutation, kind of like sickle cell.
No, I never said there's a health downside.
Well, the thing is,
because it still does the same function that the original gene does. It has an added function to it, though.
But what I'm saying,
it still does the function. So I mean, it's like saying, the thing is this is going. You're trying to make this seem like it doesn't do the original function it was meant to do.
Well, I understand it does.
Because it does.
But it's sacrificing some specificity to do so. But it's also, even if I concede that it's a truly beneficial mutation, it's really still just an amplification of a pre-existing protein. So there's nothing novel or meaningful, because remember, in our model, we predict that pre-existing heterozygosity.
Well, the thing is about mutations is that mutations that are beneficial are still beneficial, regardless, even if you do lose a little bit of the function of the original one, the original thing.
Well, I think it comes down to, because I want to hear your answer, because we did talk about genetic entropy a little bit earlier. How are you going to stop this accumulation of these near neutral deleterious mutations over time? Because obviously, if natural selection can't select against them, because I agree, natural selection is going to amplify, like we're saying with this pre-existing protein, it's going to amplify the most beneficial mutations, but at the end of the day, it's going to get rid of the most detrimental mutations.
Hold on. What do you mean by amplify?
So, for example, if we get a truly beneficial mutation, say like this one, natural selection is going to select for that, amplify it, and pass it. Obviously, it's going to pass on through the population, right? Now,
I don't understand what you mean by amplify it.
Okay, like for example, in this melanoprotein example, this pre-existing protein, the function of it was amplified because now it has this new function, this new trait based on that pre-existing protein or gene. So that gene, that trait, pre-existing, it's being amplified. I mean, you don't agree that we see amplifications of pre-existing genes and proteins?
What I'm saying is I don't understand what you mean by natural selection will amplify it.
Well, like what happens if we do see, because obviously natural selection acts on the phenotype. It doesn't act on the genotype, right? It doesn't just recognize a single nucleotide. It has to choose the whole organism, every single nucleotide, or nothing at all.
Do you have a source for that?
Well, okay, I'm going to ask you a question. Does natural selection act on the phenotype or the genotype? Go ahead.
Natural selection?
Yeah.
Natural selection is just a process.
That's but natural selection, it's not some, natural selection is not a god. It's not a deity. Natural selection acts on the phenotype. It doesn't it doesn't recognize those near neutral mutations. I mean, just Google, just write, because you can even see this on Quora, Scientific American, just write, does natural selection act on the phenotype or the genotype? And it will tell you right out that it acts on the phenotype. And that's a huge problem for evolution, because natural selection is now, and if you want to Google it as I'm explaining this, natural selection is just choosing between mutant and more mutant. So if we had 100 people in the room and you killed off 50% of it, you're still left with 50% of people who are still more mutant than the last. So at the end of the day, those accumulation of mutations.
What are you talking about when you say mutants and more mutant? There's no. If you have a mutated gene, that mutated gene in the next generation doesn't somehow get more mutated. There's no such thing as more mutated. I mean, this is the thing, is like there's no process. There's no thing that says that something goes from mutation to super mutation. I'm not saying it goes from super mutation, but if you look, just like how rust slowly builds up on a car over time, it's unnoticeable, it's invisible. You can change the brakes, you can change the tires, you can change the windshield wipers, but you're improving it, but over time it's overall it's degrading. Eventually that rust is just going to spread and take over, kind of like these near neutral mutations that go unnoticeable. Because how does natural selection select against something that's unnoticeable? Like, how do you explain and how do you rebut that problem? Go ahead. How do I rebut what problem?
Okay, so a near neutral mutation is a mutation that is deleterious, okay? But because we know that our genome is polyfunctional, okay? So, I mean, look at protein moonlighting, for example, that has recently been found out. I mean, a protein that has multiple functions, it's almost like a multifunctional tool. Now, if there's a mutation on a gene that has function, it's going to be deleterious. But if it's neutral, so if it's near neutral, selection can't select for or against it, so it builds up and accumulates in the population. So I'm asking you, how does natural selection stop this?
Okay, so in other words, what you're saying is like different eye color is going to cause us to die?
Well, no. Obviously, I agree that an example of a neutral mutation can be.
Look, the thing is neutral mutations occur, and it just means that it's not necessarily a good or bad thing. It's not going to delete anything. It's not going to harm in any way, shape, or form. That's what makes it a neutral. And you are saying that these neutral mutations are going to cause the body to slowly, like, deteriorate? Like, all these neutral mutations will cause things to completely just be done away with over time, and that I need a source for that, because that just sounds like nonsense.
Okay, so
to be honest,
so we can agree that neutral mutations happen. We've agreed that they're neither beneficial nor detrimental. Okay, so they build up.
But you're saying that they're deleterious, which means that they are harmful, but they're not.
Okay, is a single rust molecule on a car, is that harmful to the car?
The thing is this, your analogy is terrible when it comes to the human body, because like I said, eye color is a mutation, and it is a neutral mutation, and it's not going to cause over time the population of creatures to die off because of eye color.
Well, we're talking about on a genomic level that if there is any type of change, whether it's a point mutation, whatever it is, on a nucleotide, on a string of nucleotides, it's going to be deleterious to some extent. And I understand you don't like the analogy, but if I look at an encyclopedia, say on a computer that has a virus that say randomly swaps, switches, deletes, inverts letters over time, for a while there would be, just like you're saying, there would be almost no noticeable effect. But over time, it starts to contain more and more errors until
this is another terrible analogy, because I don't see where in anything in the human body is deleting and swapping letters and all this other stuff that you are using. I don't see how anything of the sort is happening. So
well, because what I'm saying, and we might be able to agree to this, unless mutations do cause a noticeable reduction in reproductive rates, the organisms that carry them will be just as successful in leaving offspring as all the others. But in other words, if the mutations aren't bad enough, selection can't see them. Therefore, they cannot eliminate them. The mutations accumulate. That's what's known as genetic entropy, and that's a prediction of our model, and that's what we see.
And it's wrong. It's wrong again. Genetic.
You're not showing us how it's wrong, though. You're not showing us
because you are making up nonsense.
Well, so if we look at the ENCODE project, right? They examined 1% of the non-coding DNA, of the human genome for possible function, but they found that at least 80% of the non-coding DNA was transcribed into RNA. That suggests function, okay? So the trajectory of discovery favors genome-wide functionality. So what I'm saying is, if this trajectory of genome-wide functionality is true, any mutation, whether neutral, positive, beneficial, on a gene that is functional will be deleterious to some extent, because it's going to affect the message overall. Because obviously you're appealing to time. Let me ask you a question. If I told you Mary Poppins could fly rapidly through the air with her umbrella, would you say that's kind of silly?
I don't know what that has to do with what I just said.
What if I told you Mary Poppins could fly slowly through the air with her umbrella? Does that make it more believable now? Does that make it more science?